Okay, sir.
Madam, good morning.
Good morning, sir.
Madam, good morning. Am I audible?
Yes, sir. Yes, sir.
Morning, sir.
Sir.
Namaste, sir.
Sir, hearty welcome, sir. Principal, sir, Dr.
Hearty
Y.
welcome
Satyanarayana,
to
sir. Please welcome, sir.
sir.
Join here. गुड मॉर्निंग कर देते हैं सर। गुड मॉर्निंग कर देते हैं। रेस्टोरेंट भी नहीं करा है।
Good morning, sir. Madam, join ayyara, sir.
చూస్తున్నాను మేడం జాయిన్ అయ్యారా చూస్తున్నాను.
గుడ్ మార్నింగ్
సార్ గుడ్ మార్నింగ్.
Good morning.
దాస్ సార్ గుడ్ మార్నింగ్. ప్రిన్సిపల్ సార్
ప్రిన్సిపల్ గారు గుడ్ మార్నింగ్.
గుడ్ మార్నింగ్ సార్. గుడ్ మార్నింగ్.
సార్ గుడ్ మార్నింగ్ అన్న.
గుడ్ మార్నింగ్ అన్నగారు. వెల్కమ్.
Can sound వస్తుంది.
మేడం చూడండి, మాధురి మేడం జాయిన్ అయ్యారా చూడండి.
Okay, sir.
కీనోట్ స్పీకర్ మేడం జాయిన్ అయ్యారా ఒక్కసారి చూడండి మేడం.
No, sir.
మేడం జాయిన్ స్టార్ట్ చేయొచ్చు.
Okay, sir.
మీకే పంపించినట్టు నా క్లాస్ ఆన్ చేసుకోండి.
నా క్లాస్ ఉంది, క్లాస్ తీసుకో.
నా క్లాస్ సేమ్ నేను అన్నట్టు
చూపించు.
Good morning, Bhikshumay sir, Vishnu sir.
మేడం గుడ్ మార్నింగ్.
Bhikshumay sir, mute చేసుకోండి కొంచెం.
ఓకే ఓకే మేడం.
ఏం చేస్తున్నావు తిరుపతి?
మేడం జాయిన్ అయ్యారా? శ్రీవల్లి మేడం గారు.
Sir.
మాధురి మేడం జాయిన్ అయ్యారా?
No, sir.
చూడండి.
पासवर्ड ये उसको उसी से कीजिए तो मीटिंग आईडी
సార్ కాల్ చేస్తా ఒకసారి.
చేశాను, జాయిన్ అవుతున్నా అన్నారు.
Yeah.
Yes, sir. JDC Gambirapet, please mute yourself. Madam, sir, Madhuri madam has joined.
Namaste, madam. Namaste, madam.
Namaste, madam. How are you?
Madam. I'm just sharing the screen. I'm trying to.
No. First madam. So, madam, start చేయండి madam. Srivani madam, start చేయండి.
Okay, sir. Is my voice audible, sir?
Yes, your voice is audible, madam. Please.
Okay, sir. A very good morning to everyone with great pleasure. Success is not the key to happiness. Happiness is the key to success. If you love what you are doing, you will be successful. I am Srimati B. Srinath, Professor of Botany, Government Degree College, Gambirapet. I take this opportunity to welcome principals, respected resource persons, all dignitaries, research scholars, assistant professors, lecturers, teachers, students of various colleges and various departments, participants from various colleges and various states of India to this webinar. Today we are conducting a webinar in association with Government Degree College for Women, JAGTIAL, on recent trends in molecular biology by Professor V. MADHURI.V. This program is aimed to give some basic and extensive information regarding recent trends in molecular biology. It is my pleasure to welcome Dr. Y. Satyanarayana Garu, Principal, Government Degree College for Women, JAGTIAL, and Sri P. Das Garu, our beloved Principal, Government Degree College, Gambirapet. Now, I wholeheartedly welcome our resource person, eminent keynote speaker, Dr. Vaja Madhuri Garu. We welcome you, Madam Ji. I sincerely welcome Dr. Masroor Sultana Garu, Vice Principal, Government Degree College, Women, JAGTIAL. I heartfully welcome Dr. Harjot Kaur Garu, IPEC Coordinator, JDC Women, JAGTIAL, and I heartfully welcome Dr. B. Bhikshamaya Garu, IPEC Coordinator, Government Degree College, Gambirapet. I also welcome with great pleasure Sri G. Chandraya Garu, Assistant Professor, Head Department of Botany, Government Degree College for Women, JAGTIAL, and Sri A. Shankaraya Garu, Assistant Professor and Academic Coordinator, GDC Women, JAGTIAL. Our senior faculty, Sri P. KRISHNA Garu, Assistant Professor of English, Government Degree College, Gambirapet. Last but not least, our organizer, I wholeheartedly welcome Dr. Padala THIRUPATHI Garu, Assistant Professor and Organizing Secretary of this program. I extend my warm welcome on behalf of Department of Botany, GDC Gambirapet, and Department of Botany, JAGTIAL. Now I request Dr. Satyanarayana Garu, Principal, Government Degree College, Women, JAGTIAL. He who made his mark in the field of administration by his sincerity, regularity, dedication, and devotion in his duties. I congratulate you, sir, for achieving NAC V++ grade. We all are inspired by your teamwork, sir. Please, sir, convey your message regarding this program. Sir, please, Satyanarayana Garu.
Thank you, madam.
Thank you, sir. Please, sir.
Heartily welcome to all the dignitaries of the today's seminar on recent trends in molecular biology on behalf of GDC Women JAGTIAL, reaccreditation by NAC with B++ in association with GDC Gambhirapet. We begin with, I congratulate all the organizing committee, conveners of both colleges, Chandre Garu and Srivalli Garu, and organizing secretary, Padala Tirupati Garu, and co-conveners also of the webinar from both the colleges. Convey my warm wishes. Welcome to our keynote speaker to today program, Dr. Baja Madhuri, Assistant Professor of KRISHNA University, Machilipatnam, Andhra Pradesh, and beloved Principal of GDC Women, GDC Gambhirapet, Pitla Das Garu, and other aspirants. Molecular biology, it deals with the function of microcalculus like proteins and nucleic acids, which are essential to life. There have been a number of major advances in molecular biology in the past few years. So we selected this topic for the advantage to the society. Thank you, sir.
Thank you for your cooperation. Thank you very much. Now, I request our young and dynamic principal, he who puts his soul and heart day and night for developing our college. Sir, please speak few words about this program. Please, Das sir.
Thank you, madam. Good morning to all. On behalf of GDC Gambhirapet and GDC Women JAGTIAL, I welcome you all. First of all, I congratulate the Department of Botany of both colleges for arranging this national level webinar. I am very much glad to be part of this webinar. I am very thankful to our keynote speaker, Dr. Vaja Madhuri Garu, Assistant Professor, KRISHNA University, Machilipatnam, for accepting to be today's speaker. I think today's topic, recent trends in molecular biology, is very much useful and relevant for all the science students, faculties, and scholars. Molecular biology can be accepted to be a mainstay in the manufacture of chemicals, energies, medical, consumer products, agriculture, and food industry. I convey my best wishes to organizing secretaries and conveners and co-conveners. Thank you.
Thank you, sir. Thank you for your support and encouragement. Now I request Vice Principal Dr. Masroor Sultana a few lines about this program. Please, madam.
Thank you, madam. Thank you very much.
Thank you, sir, madam.
Okay. 1 minute, ma'am. 1 minute. Okay. Respected principal of our college, Dr. Y. Satyanarayana, sir, and principal of GDC Gambhirapet, Sri P. Das, sir, convener Sri G. Chadra, sir, from GDC Women's JAGTIAL, and Srimati B. Srivalli, ma'am, from Gambhirapet, co-convener Sri A. Shankaraya, sir, organizing secretary Dr. Padala THIRUPATHI, sir, keynote speaker Dr. V. Madhuri, ma'am, assistant professor, KRISHNA University, Machilipatnam, Andhra Pradesh. Participants and my dear students, good morning, everybody, and welcome to this national webinar on recent trends in molecular biology, organized by the Department of Botany and NSS of GDC Women's JAGTIAL with the Association of GDC Gambhirapet of Telangana State. The study of chemical and physical structure of biological macromolecules is known as molecular biology. I think these type of webinars are very useful to our students. They mostly belong to rural areas. though they are very talented, they lack knowledge of research work. With proper guidance and motivation, they can definitely achieve their goals. So such webinars create interest in subject. Hence, and I appreciate and congratulate all the faculty members who are involved in this program. And I wish for the success of the webinar. Thank you very much, ma'am.
Thank you, madam. Thank you very much. Thank you for supporting and encouraging us. Now I request Dr. Harjot Kaur Garu, IQAC Coordinator, GDC Women JAGTIAL. Madam, please, few words regarding this webinar.
I hope Madam is not joined. Madam will join later.
Okay, sir.
Madam, please proceed.
Now I request Dr. P. Bhikshamaya Garu, IQAC Coordinator, GDC GOVT.DEGREE. Are you there, Bhikshamaya, sir?
Okay, madam. Okay.
No, sir. Sorry, been busy with NAC work. Now I request our senior faculty, P. KRISHNA Garu, Assistant Professor of English. Sir, please, few words for this webinar.
Good morning, professors. Good morning, organizing secretaries. Good morning, principals of both colleges. Good morning, the participants. It's a great opportunity not only for the students but also students, the faculty members to have very enlightened thought about this molecular biology. I will be talking about biology and even get exposed to biology everywhere in the classes and outside of the classes. But they have to be given knowledge regarding what are the recent trends. What are the recent trends in the molecular biology? And really, I appreciate the hundreds of the good colleges who venture themselves to come outside and take these students to the recent trends in the vibrating of the subject like this. And this is the thing that is required by the Commissioner of Teacher. Not only teaching and limiting the students to the classroom, but also allowing them to know something about the recent trends. Really, it's a great opportunity for the students, really, those who are getting admitted into this webinar. So this kind of seminars are going to enlighten everyone. I really thank you very much for this opportunity. I also now, though I belong to languages, but I really feel very happy to know something about the molecular biology. What kind of the wonders that are being created in the living organisms, maybe in the humans, maybe in the animals, maybe in the plants also. This is something extraordinary. I appreciate, and I appreciate our civil medical. I appreciate our THIRUPATHI Garu for initiating this kind of program. This kind of programs are much needed in the present scenario. Thank you very much for having this one. Let us tell you again.
Thank you, Krishnamurthy Garu. Thank you, sir. Now I request Sri G. Chandraya Garu, Head Department of Botany. Please, sir, few words. Chandraya Garu. Are you there, sir? Chandraya, sir?
Okay, madam, sir will join later. Okay. Please you proceed.
Okay, sir. Okay. Now I request A. Shankaraya Garu, co-convener and academic coordinator of Government Degree College Women for JAGTIAL. Sir, please share your message.
Second, madam. Sir is very busy. I hope.
Okay,
Madam,
okay, sir. Okay, now I request Dr. Padala Tirupathi Garu. Please introduce our esteemed keynote speaker, Dr. Vaja Madhuri Garu. Over to Tirupathi sir. Sir, please continue the session.
Thank you, Srivalli Madam Garu. So,
thank you, sir.
We are wholeheartedly welcoming all the dignitaries and delegates of our country and respected principal sir and vice principal madam, IKC coordinator and academic coordinator, and also the principal GDC Gambhir, our beloved brother, Dr. Pitla Das Garu, and IKC coordinator Dr. P. Bhikshamaya Garu, and senior faculty of GDC Gambhir, wholeheartedly welcome you, sir. Now, myself, Dr. Padala THIRUPATHI. My immense pleasure to introduce one of the eminent professor, keynote speaker, MADHURI-V garu, of Biosciences and Biotechnology, KRISHNA University, one of the esteemed university, Machilipatnam, Andhra Pradesh. Madam has a huge experience of her professional career, more than 21 years of PG teaching, and even though along with PG and UG teaching 5 years, Madam has 5 years experience. And at the same time, industrial experience. Madam has industrial experience also more than 2 years. Madam has worked as a junior scientist, Leela Nutraceuticals, Vijayawada, and Madam academic career, Head Department of Botany, Andhra Loyola College, Vijayawada, Head Department of Biotechnology, Andhra Loyola College, Vijayawada, teaching as Acharya Nagarjuna University, Guntur. Now, presently, Madam has working as an assistant professor, KRISHNA University, Machilipatnam, Andhra Pradesh. Madam academic profile. Madam completed her UG degree in 1899. Madam has completed her PG, topper of the Central University, 1991. And Madam completed through University of Delhi in plant tissue culture biotechnology in 1993. The title is Shoot Apical Meristem Culture in Capsicum Annum. And Madam has completed her in medicinal plant research biotechnology, Acharya Nagarjuna University, Guntur. Title Comparative Molecular and Bioactive Studies on Selected Species of Calma, RBR, and Borosia, WIT, and ARN 2014. This is about Madam academic career. And Madam is also published a number of papers and published a book chapter. A book chapter title is Evaluation of Depigmentation Activity of Xylea, Xylocarpa, and its phytoconstituents is very well written and has been accepted for publication in edited book titled Advances in Biotechnology and Biosciences, Volume 6 in 2020. Madam has conducted a number of conferences, webinars, and also Madam published a number of reputed national and international journals. Madam has this huge experience is very useful for today's webinar, national webinar on recent trends in molecular biology. This is also very useful for all the UG students and also PG students of the biotechnology and the life science stream, and also it's helpful for all the faculty members, those who are working in the various degree colleges and PG centers of the various universities of our nation. So we are wholeheartedly welcoming you today's keynote speaker, Professor MADHURI-V Madam garu. We are wholeheartedly welcoming you, Madam. Please start the session. Madam, please unmute. Madam, please unmute yourself.
Respected dignitaries, whoever present here, respected Principal of the GDCW, JAGTIAL, Dr. Y. Satyanarayana garu, respected Sri Pitla Das garu, Principal of GDC, Gambhirpet, and respected conveners of today's national seminar, Sri G. Chandraya garu, B. Srivalli Madam, A. Shankaraya sir, and also today's organizing secretary of today's one-day national seminar, Dr. Padala Tirupati garu, and other faculty members and the vice principal of college, Manasur Sultana Madam, and other faculty members, my dear students, research scholars, very good morning to one and all. So today I am going to talk about, I'm going to share now the screen, please see all of you. So today I am going to talk about crop improvement, how the crop improvement can be taken place. So crop improvement is very much important for everybody because with respect to the increasing the crop yield, nutritional quality, and if you want to grow them in branching, high branching, dwarfism, and maturity, and the duration level reducing, pest resistance, disease resistant, whatever may be, all these things come under crop improvement characters. This crop improvement can be achieved by simple methods or by plant breeding methods like mass selection, backcross selection, self-pollinated, cross-pollinated, and also vegetative propagated plants. There are several methods are there in plant breeding. Next level comes to the genetic engineering method, where you can do the crops genetically modified in such a way you can improve the crop. So genetically modifying means you are creating a plant with desirable characters. Obviously, you concentrate on the improvement of a crop. The next comes to the after transgenics marker assisted selection. So you can select the plant with a particular marker, molecular markers like RFLP, RFPD, all those things, and you can select those plants also. And the next, now it recently developing area is RNA interference. This is also one of the mechanism, one of the method that is applied for the crop improvement. So what is the role? What is the mechanism? How the applications of RNA interference you can observe while doing crop improvement. So not only this crop improvement helping the plant to be improved, and also one can earn money also. So that we can call it as biotech's $1 billion breakthrough. So this RNAi for the last decade, it is growing a lot. This is a growing field in the molecular biology. So stepwise, we will go one after the other. So here, as we know, the production rate of the plants is getting decreased, the population rate is increasing, rapid urbanization, industrial belt, all these things, soil erosion is taking place, climate getting changed, pollution is taking place. So natural resources are getting depleted, land, water bodies are getting depleted, and you need to improve the seed, you need to improve, or you need to develop disease resistant and pest resistant plants. And also at the same time, the plants must contain effective plant nutrients. They must be resistant to abiotic stress. They must be resistant to environmental problems, whatever occur day to day. So you need to go for sustainable technology, the technology which is useful to maintain the plants yesterday, today, and also tomorrow. So you have to invent for that and concentrate more on that subject so that you can get sustainable rate in the crop improvement. So why we should go for the first thing, simple farming. The second level, plant breeding. A third thing that comes is transgenics, genetically modified crops. And the next thing, RNAi. So why we should go for GM crops, genetically modified crops, genetically modified foods? What is the need for that? So there is a challenge for that. The population is growing day by day. Human hunger diseases, malnutrition is playing, malnutrition is high level in this. So there is a global demand is there for increasing the food products, for improving the quality of the crop, for improving the food production. So the technology options are plant breeding, then genetic engineering. So there is a need for second green revolution. So then what you see here, these there are agrobiotechnology, biotechnology. There are two terms are there. Biotechnology means biology, biology field. So there is a basic needs for food, clothing, shelter, health, and safety. All these must be improved. In agrobiotechnology means in agriculture, in crop fields, you need to improve the quality, you need to improve the pest resistance, disease resistance, bacterial resistance, pathogen resistance, abiotic stress resistant, and also all nutrition important characters. Not only that, visible characters also. For example, here, but there are some characters human desires, but they are not available in the fields. So what you have to do, you have to create, you have to go for the technology, new technology. So here the fruits, normal banana fruits we take, they don't have vaccines. But if you produce the fruits with vaccines, so blue roses and also cotton, so something like that, you have to do that. So there must be a plant with a desirable character. There is a plant. What is a plant? This is a banana. A desirable character is having vaccine. Simple, by eating banana, you can get vaccinated. And also similarly, it is rare to get blue roses. The blue roses, you can see pink roses, white roses, but people want to see blue roses. So there is a technology. And also blue cotton. These simple things. So technology is for this. So the grains with improved nutrition. So these are all strawberries, genetically modified strawberries. So these are all the plants which are improved with their qualities or color, or maybe showy appearance, or visibility, or nutritional quality, whatever may be, you are changing the character. So there is one industry that is name is Gentech. Recently, genetically manipulated corn they developed. So what if you eat this corn, fat is burned, so obesity will be reduced. So that's how they concentrate on one particular character. Simple, by eating this corn, one can reduce their fat. So the industry that developed is Gentech industry. So organism who eats this corn, he starts losing his fat deposits. So like that, the technology is playing important role. So after plant breeding, you can see they're making a transgenic approach. Transgenic means transfer of a gene, a foreign gene, into a plant where it is not there. So you can transfer a particular gene using a binary vector, that is agrobacterium mediated tumefacts. Now all of you might be knowing about agrobacterium mediated transfer. So you are transferring a gene using a vector. So you are using suspension culture. What is suspension culture? What is inoculation? What is a vector? What is the gene gun? How do you produce all those things once? See here, what you have to do. If a gene of interest, that means a particular gene is responsible for a character. So that gene of interest, first you have to isolate. And that gene of interest must be placed in a vector. A vector is a vehicle. A vehicle is the one which carries the genetic information from one place to the another place. So in this vector, you are tagging your gene. And through the agrobacterium you transferred it. And this agrobacterium is inoculated to the hypocotyl region. You know what is cotyledon? Cotyledons of the first leaves. Below the region you can find hypocotyl. Above the region you can find epicotyl. So at that epicotyl region, you inoculate with the agrobacterium and you place it on MSC medium. And then slowly callus is formed. And the callus, if you suspend in the liquid medium is the medium that is a morass. You can call it as suspension medium. In this suspension medium, just keep it and put it in the shaker. You will find the cells and also sometimes embryos. Embryos means those embryos, heart-shaped, torpedo-shaped, globular embryos you can find. These embryos, they will give rise directly to the shoots. The shoot induction medium is supplemented with the cytokinins, and the root induction medium is supplemented with oxins like naphthalene acetic acid, indole acetic acid, R2,4-D, sometimes with permutations and the combination with the auxins, and it may change from species to species. You can find the shoots. After developing the shoots, you have to go for the root induction medium. Thereby, you will get a plant, and this plant is called transgenic plant with a desirable gene. Previously, the plant is without that gene, but after introducing that gene, that is called transgenic approach, this plant now having the gene of our interest. So like that, the corns, like that, all those blue roses, those are all developed. So for everything, it takes so many years for doing research, for the development, for getting commercialized, that means marketing and earning money. See how many years that is taking place for the trait generation, optimization, and also you have to form a concept or a model plant and then testing and then field trial, whether that plant can survive in the field in the normal conditions or only it can survive in greenhouse effects. Then finally you have to go for commercialization. Like that, it takes so many years. Now, recently, I think 2008 to 2003, something like that, RNA interference, there is another technology, there's a sustainable technology where one can improve the crop character, where one can produce disease-resistant plants, whereby using, you can do virus-resistant plants, worm-resistant plants, where you can destroy the viruses, you can destroy the bacteria which will attack the plant. So that technology, that sustainable technology is now called as RNA interference. RNAi, I means interference. You know, all of you know what is the central dogma. So gene is a segment of DNA. The DNA is the double-stranded DNA, out of which one is the target DNA. From the target DNA, you will get hnRNA. So how much ever length of DNA is there, you can find the similar length of hnRNA. hnRNA means heterogeneous nuclear RNA. It consists of exons and also introns. The introns are cleaved off and the processing takes place. Thereby all exons make up mRNA, that is the messenger RNA. The sequence of nucleotides, ribonucleotides, determine the sequence of amino acids in polypeptide. Thereby you will get a protein. So what is the central dogma? DNA to mRNA by transcription, mRNA to polypeptide chain by translation is a central dogma. In this central dogma, these polypeptides make a protein and all enzymes are proteins. The proteins include structural proteins as well as functional proteins, which will create the phenotype. From the phenotype, you can say this particular protein is there. To go back, that particular DNA is there. So if you silence that particular DNA, mRNA is silenced. If mRNA is silenced, no more protein. If no more protein, that particular phenotype cannot be seen. This type of silencing is called gene silencing. So what you are doing, you are suppressing the gene, not to go for mRNA. If it is not, mRNA is not formed, that polypeptide is not formed, so that phenotype is stopped. That is called gene silencing. It is of two types. Gene silencing you can do at a transcription level. Gene silencing you can do at a translation level. Transcription, as you know, from DNA to mRNA. Translation is mRNA to polypeptide. So here, these are the basic things that you should be in your mind before understanding the technology. The technology of RNA interference, how it is useful, how you can do research, even you can do, if you learn the technique, you can do miracles in the lab and you can produce whatever you want in plants. Only thing is you have to practice the technique and the basic knowledge you have to know. So here today I'm going to deal with that RNA interference. As you know, RNA means mRNA. mRNA means messenger RNA. RNA is always a single stranded. RNA consists of ribonucleotide sequence. Okay, so this RNA, how it is RNA interference.
Excuse me, madam. Please, excuse me, madam. Sorry for the interference. Sorry for the disturbance, madam. So this session will going to wind up this session. Please, all of you, all the delegates, personal requesting you, please join through same link.
Same link, okay.
Madam also, personal, madam, all of you join through same link after wind up this session.
Okay. Okay, sir. Thank you.
So previous. Okay.
Okay, madam.
So what is RNA interference? Interfering what? It is interfering the gene. What is the gene? The gene which is responsible for certain character. So it is silencing that particular, it is blocking that particular gene function. So how it is doing? So there is one sequence of RNA which exactly matches to the target gene. So what will do? This goes and blocks the DNA gene function. So since since science named it as a breakthrough of a year, because previously there was a simple farming followed by plant breeding, followed by marker assisted selection, followed by genetic engineering. All these methods are leading to crop improvement. But now another technology, that is RNA interference, is a methodology that is useful for improving the crop. So it is a breakthrough of the year, and the Fortune magazine hailed it as a biotech's $1 billion breakthrough. That title given in 2003. So it has gained prominence in so many aspects. If you see in human beings research and also some disease related things, also it gained many functions. But only thing for this is the particular gene must be known, the target gene, its structure and function must be available to all. So RNAi has the potential to become a powerful therapeutic approach towards targeted and also personalized medicine. Nowadays, the same approach, the same technology is applicable also to agriculture. That is called agri-biotechnology. Agricultural improved characters, crop improved characters. For this RNAi is playing very important role. So it provided a way, this RNAi, that is this RNAi, it is playing an important role to control pests and also diseases. So pests, diseases, pathogens, bacteria, and fungi, which are spoiling the plants, they are getting destroyed because of this approach. Thereby, the yield of the crop is increased. So the scientists who are doing this RNAi research, they have developed novel crops. They have developed a tobacco plant with nicotine free. They have developed a peanut plant with non-allergic. As you know, peanuts, some people feel allergic. They developed decaffeinated coffee. They developed nutrient fortified maize. So all these things are developed by so many scientists who are working on RNA interference. What is RNA? Once again, I'm telling, from DNA you will get messenger RNA. From messenger RNA we will get polypeptide. Some polypeptides make a protein. Some are structural proteins, some are functional proteins. All enzymes are proteins. This will show the phenotypic effect. This will show disease resistance. This will show crop improvement. This will show blue rose. This will show a maize with a certain nutritional quality. So that character is obtained only because of their genetics. So what you are doing using this one. So how this RNAi discovered in plants? What is the plant? The credit of the plant goes to the plant Petunia. Petunia is a flower with excellent purplish color. You feel very happy to see that one. Very pleasant color it has. So scientists started thinking, okay, what are the things that one can improve the crop is? Simple farming, just by hybridization or self-pollinated methods, cross-pollinated or plant breeding methods, then genetic engineering method, slowly they are doing. So while doing this genetic engineering methods, scientists, what they thought, okay, we'll also improve this purple color, more purple color, which is more pleasant to our eyes. So we feel more joy after seeing those flowers. So what they have done, they started doing or introducing the gene that is responsible for purple. So they introduced this gene into the plant. They're expecting, okay, all the flowers came, but instead of more purplish color or deep purple color, what they got? They got some are white in color and some are variegated in color, that mix of purple and variegated with the spots. But they didn't get dark purple, what they are thinking of, what they are designed. So they don't know the mechanism that is behind it. They thought, okay, they introduced the gene that is responsible for purple color and expecting, yes, that same color they will get. But instead of that, while doing that experiment, the reverse has taken place. White color obtained, variegated obtained, light color obtained, which is not pleasant to them. So they, at that time, they don't know any phenomenon such co-suppression or RNA interference, such type of things are there, are existing. At that time, they don't know. They just observed that character. So that is called, they thought that it is an unknown mechanism. The mechanism is not known. Even though they have introduced the transgenes, what is happening there? Silencing is taking place. The gene became silenced. Gene is not showing its expression. Gene is not showing mRNA. Gene is not showing mRNA to protein. This is responsible for anthocyanin pigment or by purplish color. It is not showing. So how this silencing is taking place? At that time, they don't know. That's how the discovery will start. The hidden mechanism, they started thinking. So in another research, gene silencing was also observed when the plants are infected with genetically engineered viral vectors. You know what is vector? Vehicle. Genetically engineered means if RNA gene is there, vector DNA is there, and its promoter regulator sequence is there. Now the vehicle is ready to get injected into a plant. That is genetically engineered. That was the research before RNAi. So that engineered one also, they are unable to produce. They are unable to produce what is their important or desired character. So at that time also they don't know what is the mechanism behind. So then the mechanism came into the discovery, that the discovery will start now. So there are two scientists, they have done excellent research and excellent presentation and the result. Thereby they got Nobel Prize also. That credit goes to Andrew Fire and also Craig Mello. These two scientists, what they started thinking, what is the mechanism behind it? Instead of getting elevated of the pigment production, that pigment production is getting decreased. Instead of getting the desirable character, the opposite is taking place. How it is happening? So the credit now goes to Andrew Fire and Craig Mello, who got a Nobel Prize in 2006 in the field of physiology and medicine. They called that phenomenon as RNA interference. How is? So they discovered, they injected a double-stranded RNA. As you know, DNA is double-stranded. Only one of the strand acts as a target for the synthesis of a single strand of mRNA. But here, what is that one? RNA. RNA is double-stranded, and you are injecting that into the Caenorhabdis elegans. Caenorhabdis elegans introduced, they both of them introduced or induced or injected into that organism. What happened? The silencing has taken place. Silencing has occurred. Whatever expression you are expecting, not getting. So the double-stranded RNA is entered into another path. That path that is called a mechanism where this mRNA that is responsible for the protein synthesis is getting degraded. From outside they injected double-stranded RNA. Not this is endogenous, exogenous. From outside, thereby silencing is taking place. This phenomenon is called RNA interference. The similar type they observed in petunia. Petunia, what? Just in the previous slide, I explained about purplish color, anthocyanin pigmentation. Instead of getting more pigment, they got white and also variegated. Why? Because there is another path called co-suppression pathway. Co means side. Suppression, suppressing. We will discuss in next slides also. Virus-induced gene silencing. The virus-induced, that means a virus that is carrying a foreign particle inducing the gene silencing. We revealed that these processes led to the double-stranded RNA, thereby interference. So, because of the presence of these pathways, double-stranded RNA is getting accumulated where inside an organism. Thereby interference is taking place. So what is the starting point for any RNA interference here? The starting point always is a double-stranded RNA. Whether it may enter through the vector or genetically engineered or any other thing. If it enters means it creates a path. So that's how the discovery is led to the gene silencing. That is a phenomenon called RNA interference. We can call it as co-suppression in plants. We'll discuss in details. In addition to its gene regulation, that means silencing the gene, thereby stopping the expression. Gene silencing means what? Gene to mRNA, mRNA to protein is stopped. That is silenced. So in addition to those roles, RNA interference is also used as immune response to infection. So if any infection has taken place, immunity also, that also role we'll study later. And natural defense mechanism, jumping genes, viral elements, and specific type of bacteria also trigger. So for all these things, they trigger the synthesis of double-stranded RNA, they trigger RNAi pathway synthesis, they trigger the stopping of gene expression, thereby the gene is not expressed mRNA, not formed polypeptide, not formed proteins and enzymes are not formed. Not formed is the term leading to suppression or silencing. So now, what exactly is happening? What is the mechanism behind it? Why always you are saying double-stranded RNA is creating a problem? So we'll see the stepwise. So what are the steps involved in it? So what is central dogma again? DNA to mRNA, mRNA to polypeptide. Here, double-stranded RNA. So just entering double-stranded RNA into a plant. Just imagine, into a plant, a double-stranded RNA is entering. How it enters? It may be through a virus, it may be through a vector, or it may be through a genetically engineered an organism, any other thing. But a double-stranded RNA is entering into a cell. So now what is happening here? Double-stranded RNA is inside the plant body. Now, automatically, a dicer is a protein, enzyme, dicer. What is the nature of dicer? You know what scissor does. Scissor, kattira. What it does, scissor goes and cuts. If it is in double-stranded form, double-stranded cuts will be formed. So what I'm saying, double-stranded RNA entered. Now a dicer enzyme is recruited automatically, and thereby this dicer goes and cleaves the double-stranded RNA. Double-stranded RNA ela unte alage cut chestund. So what will happen? 20 to 25 base pair long fragments. These are called siRNAs. RNAi is RNA interference. DICER, dicer means enzyme. Enzyme role is scissor role. Scissor what is doing? Double-stranded DNA, RNA is cut. Now, a small fragment, just imagine a thread. So it cuts into pieces. So small fragments you will get. This fragment length is of 20 to 25 base pairs. So RNA means ribonucleotide base pairs. DNA means deoxyribonucleotide base pairs. So here base pairs means ribonucleotide base pairs, 20 to 25 base pair long. These fragments are generated. From where they are generated? From double-stranded RNA. So who cut the double-stranded RNA? Dicer cut that. What is dicer action? Like a scissor. Now what are the things you got? siRNAs. What are siRNAs? Small interfering RNAs are obtained. So now another protein. What is that complex called? RISC. What is RISC? R means RNA. So this RNA induces. So induction takes place, thereby itself automatically generate. That is called risk factor. RNA-induced silencing complex. This complex goes and binds between, you are having in 20 to 25 base pair long fragments. What are those called? siRNAs. So in between those two fragments, risk goes and binds. Whenever this risk goes and binds between these two, sense strand and anti-sense strand, both the strands will get separated. Okay. So once again, double-stranded RNA, dicer. So single-stranded, not single-stranded. Small interference RNA, small fragments, even though double-stranded. So there are two strands, sense strand and anti-sense. In between them, now risk entered. What is risk? RNA-induced silencing complex entered. Thereby, sense strand separates, anti-sense strand separates. Now, the sense strand, which is exactly similar to the target gene. Say for example, target gene means DNA strand. DNA strand, if you have the sequence like AGCT, this has, instead of thymine, in RNA you will be having uracil. That's the difference. So the sense strand is exactly similar to the target strand, gets degraded, and now the leftover is antisense strand. This antisense strand incorporated to the RISC and used as guide strand. So guide to the what? Guide to the target mRNA in a sequence specific manner. So what will happen? Generally what will happen? The nucleotide sequence determine the nucleotide sequence of mRNA, and which in turn determine the amino acid sequence. This is a stepwise. So what will happen? This mRNA, which is coded for amino acids, are getting cleaved. From mRNA, you should get polypeptide amino acid sequence. If mRNA gets degraded, what will happen? There will be no protein. That's how inhibits the protein synthesis. Briefly, double-stranded RNA, dicer, small interfering RNA of 20 to 25 base pairs. Small interfering RNAs, what are they? Sense strand, antisense strand. Sense strand, it gets degraded. Antisense strand remains. From where is supposed to get amino acid sequence, but this amino acid sequence cannot be formed because whatever antisense strand is getting, it is getting broken into small pieces by RISC. So that is how gene silencing is taking place by RNA interference mechanism. So this is how, so long DS, dicer cutting into bits of 20 to 25 nucleotides. These are siRNAs, RISC formation, sense and antisense separation, thereby fragments we can get. So the first gene silencing methodology also developed in some varieties which are in plant varieties with resistant to viruses. So what is happening? So some viruses like tomato spotted wilt virus, bean golden mosaic virus, you know all those things, banana bract mosaic virus, Rhizotangro bacilliform virus. So many viruses, they will come and spoil the parental plant vigorously. So thereby the crop yield will be decreased. So what do you want to think? What a farmer think? What a plant breeder think? What a genetically engineered scientist will think? Now what RNAi research doing scientists will do. Okay. Now this scientist who is familiar with RNA interference technology, he goes for this technology to develop a plant variety which are repeatedly spoiling the plant. What is that? A virus that is spoiling the plant. So for example, they have done an experiment on potato virus Y-resistant plants that expressing RNA transcripts of a gene. The target gene is here viral proteinase gene. So they successfully developed RNAi technology against the potato virus, which is spoiling the potato plant. So similar type of technology also developed for a number of viral attacks, so that now the plants are resistant to viruses, the plants are resistant to nematodes, the plants are resistant to insects, pests, and various pathogens because of silencing of the gene. Which gene? Which is causing the disease, which is spoiling the plant. So that gene is getting silenced. So no more damage to the plant. It's a direct approach. So similarly, male sterile lines. What is male sterility? All of you know. It is also one of the important, it is very important for the crop improvement. Why? The simple logic behind it. A bisexual flower always go for self-pollinated or self-fertilization, thereby the same characters repeated. But if you make the sterile of male, then what will happen? That bisexual female no more functional as male, so it acts as a female flower. Now this female flower is ready to accept the pollen from a desirable character containing plant. So the pollen from the desirable plant easily you can put it on the stigma of a plant so that it will reach and fertilize and give the result. The normal emasculation plant breeding techniques that done in plant breeding experiments. But the role of RNAi, how it is playing means the RNAi that means the RNA interference suppressing a gene that plant, even though it is bisexual, the male part is not fertile. It is not at all fertile, sterile. So the birth of the plant itself is like that it is a male sterile. So what is happening here? No more receive its own pollen. It is not ready to receive its pollen because the pollen is not functional. The male gametes are not functional. So like that, male sterile lines can be produced. How the fertility causing gene is suppressed. Male fertility causing gene is suppressed. That means male becomes sterile. Now naturally the plant receives the pollen from the desirable characters. So you see here, RNAi has also been used to generate male sterility. Male sterility means it is very, very valuable in the hybrid seed industry production. In hybrid seed industry, whatever seed with desirable character you want, you can produce. So you know what is cytoplasmic male sterile, genetic male sterile, cytoplasmic and genetic male steriles, restaurant lines, plant breeding restaurants, RS, backcross, repeated selections, all those things studied in plant breeding. Here the plant itself becomes a male sterile, even though it is a bisexual plant. How? By using RNAi technology. For example, there is a gene called TA29. We have to remember such type of names. TA29, a gene that is necessary for pollen development. If this is suppressed, no pollen. If pollen is suppressed, male is sterile. If the male is sterile, the bisexual flower behaves as a female only, not male. So the technology also developed like that. First, just by emasculation methods. That means bagging. Next, by CGMS and GMS, that is cytoplasmic genetic male sterility, genetic male steriles, plant breeding experiments, repeated recurrent selections with male sterile, male fertile, RS factors, restoring factors. There, the technology developed furthermore, Barnes system that you can see in the genetic engineering system, where you can produce male steriles. Now, here the system, RNAi system, next system, it's recent system. This system is used to suppress a gene which is responsible for pollen production. If you suppress the gene, male production automatically becomes male sterile. So, similarly in tobacco, in maize, also developed male steriles using RNAi, that is RNA interference mechanism. So if you see the simple RNAs, coding part and non-coding, how they are, this is the next biological revolution is based upon. So here you can see Sinorhabdis, here you see Petunia. The history started, discovery started with these two organisms. Successfully interference here, RNA interference. In Petunia, you can see co-suppression. So this is the next generation evolution here. So different types of RNAs, mRNA and ncRNA, means non-coding RNAs. So here, what exactly and who did the experiment? What is the credit? Where the credit goes? We'll see here once again with the details now. So here, this is a Petunia. This is this color pigmentation. This pigmentation is due to anthocyanin pigment. So anthocyanin biosynthesis metabolism will be there. So pathway will be there. So many enzymes are needed. Enzyme means a protein. The protein means from mRNA you will get protein. mRNA you get from DNA. That is the path. So anthocyanin biosynthesis is a multi-step process. So here the credit goes to Richard Jorgensen. So he is in 1990s only. He discovered gene silencing method while working on Petunia. Petunias developed some areas called hypopigmentation when transfused with a gene encoding the enzyme required for pigment synthesis. So I'm saying you anthocyanin pigment synthesis is there. So from A to B, B to C, C to D, D to E, finally from E to anthocyanin, you will get that pigment. So there are so many enzymes also there. Enzyme means protein. This protein is from DNA. So that DNA, if you make it silence, then no anthocyanin will be produced. But they don't know that they are going for hypo, and hypo means less. They wanted to do hyper, means more pigment. But hidden mechanism, what have done, co-suppression has taken place. Repeatedly inducing transgene into a plant where this plant is expected to contain more pigment. What is happening? One gene is suppressing the second gene. Second one suppressing the third gene. So what is happening? Co-suppression is taking place. Thereby, no more pigment has taken place. This is a very good example in co-suppression. That is one of the form of the RNA interference. So from outside, your transgene is, that is, you are transferring a gene. That's why it is called exogene. And it is suppressing endogene. Inside the gene, you are suppressing. So inside the gene, you are suppressing. So here, exogene suppresses the homologous endogenous thing. So endogene means what? Inside the plant, there is a gene. From outside, you are introducing to increase the pigment. But instead of increasing the pigment, it is started suppressing one to other, second one to third one, like that co-suppressing. Thereby, what is happening? No pigment. So this is called gene silencing. That paper is also published in article. This article also there. If anybody interested, can go through this article, and everybody can do the research very easily if you know the concept and only simple technique. Only thing is you have to motivate. You have to go to this type of articles. You type it Google Scholar, this article, you will get the entire article also, and this is published by these people. Co-suppression. And the same phenomenon, if it is observed in fungi, they are called it as felling. So Petunia, he is the person, the jargon son. 1990s only, no? Next credit, we were discussing about the Nobel Prize winners. Two scientists were there. So these two, Andrew Fire. So they have done their work in Rhabditis elegans. Rhabditis elegans. So they got a Nobel Prize. They have used that RNA interference, and thereby they did the experiment, and they also published an article in this journal, double-strand RNA made gene silencing. So now this double-stranded RNA, it is a post-transcriptional process. So it may occur before mRNA formation, or it may occur after mRNA formation. So like that, this is the one. So antisense RNA post-transcription means after mRNA formation, that mRNA is getting broken into small pieces, no more polypeptide chain can be formed. This is another article published by Hamilton. So you can go through and you will get some idea if anybody interested in doing research in RNA interference. So nowadays, what is happening here? This is the era of post-genomic era of plant biology. A major challenge is to determine what is the structure of a gene, followed by what is the function of the gene. Thereby, you have to study the whole plant genome. Compared to other techniques, that means genetic engineering, plant breeding, or simple farming, all those things. RNAi also offers specificity and also efficacy in silencing genes. The gene may be a single gene, or a member of a gene, or a gene belonging to a multi-gene family. So here, double-stranded RNA, in addition, the expression of double-stranded RNA with a promoter, the promoter must be an inducible promoter. This can control the extent and also timing of the gene silencing. When has to be silenced, how much has to be silenced is also can be determined by using a inducible promoter, such that essential genes are only silenced, only chosen genes at a particular stage, at stem stage, at a root stage, at a flowering stage, at embryo stage. At particular stage only you can arrest that particular gene. So you need to have inducible promoters so that at a particular growth stage, in a particular stem or a root or a leaf or a flower or any other organ of a plant, you can suppress at that particular age. So there are several ways in RNAi pathways. So many ways are there. I explained only a simple way. What is that simple way? Double-stranded RNA from small bits of RNAs, how it is happening with the risk and factors. So that is the simple mechanism. Now we are discussing about different mechanisms also. So what are the advantages are there? What are the disadvantages there? So different modes, how this double-stranded RNA is induced, maybe by virus or maybe by transgene. We differentiate all those mechanisms. So here we see different types of mechanisms. Here you can see different. Just see here what are the different mechanisms. What is the simple mechanism? We have already seen dicer, role of dicer, role of risk. Okay. Here we are seeing what is the source, how the double-stranded RNA is obtained. What is the source and how the double-stranded RNA is obtained, the source. So this double-stranded RNA creates the problem. So how it creates? What are the different types we see? So what is the source? It may be a virus. It may be a inverted repeat. It may be a transgene. The gene that is the transposon, the jumping gene avachchu, mobile element avachchu. Or it may be a foreign gene which you want to introduce. That may be a source avachchu. Then for what? For the double-stranded RNA. Source means what? From which this double-stranded RNA is obtained. So these are the sources. It may be A or B or C. It may be a virus. It may be an inverted repeat. It may be a mobile element, transgene, a transposable element. So from that, you are getting double-stranded. Red color, 2 lines are double-stranded RNAs. So double-stranded RNA, from where are you getting? Viral RNA by viral replication, from single strand to double you will get. Inverted repeat, how it is happening? Because it is inverted, break has occurred and it gets double-stranded. Transgene transposon, so transcription has taken place. Transcription means gene to transcription means you will get RNA. This is called priming. So priming means the first formed ones. So that is obtained first. This priming RNAs will become here again double-stranded. Red color indicates double-stranded. Priming means single. So here, this is how double-stranded RNAs are obtained. Next comes to the this one, transcription. Transcription, the next step, intermolecular pairing. Sometimes it also takes place. So here the mechanism includes dicer, again double-stranded RNA, dicer. Dicer role is scissors only. So it is cutting into double-stranded bits. It is energy-driven process. So you need energy. So energy is utilized. Thereby ATP is broken into ADP. So this is single-stranded ones. Now combined with risk factor, but that risk factor is silencing complex, RNA-induced silencing complex. This one is inactive now, but gets activated when again one ATP molecule is hydrolyzed, energized. So this complex get energized, thereby you will get this complex, thereby it undergoes degradation, becoming small fragments. Once it gets degraded, it is no more produce a polypeptide chain. When it is continuous, then only it has the capacity to produce a polypeptide chain. But when it is broken, no more produce polypeptide chain. And this approach is a transgene with hairpin model. Hairpin, if you see the hairpin will be like this. So hairpin model construction. So these are all the different types of mechanisms you can see. It may be by viral infection or by inverted repeats or by transgenic approach or by transgene with hairpin structure. These are the powerful inducers of double-stranded RNAs. Thereby these double-stranded RNAs are combined with the dicer and gets activated by ATP. Thereby bits you will get double-stranded bits, and these two get separated by risk factor, risk mean, RNA-induced silencing complex. Thereby this single strand becomes fragmented and no more useful for polypeptide chain synthesis. So what are the different mechanisms? What are the different types of this interference? So you can see here siRNAs, that is short interfering RNAs. shRNAs, short hairpin RNAs. And also another one is microRNAs, miRNAs. See, mi, micro. H. What is that? Short hairpin. So these are the different types, and different types of mechanisms get operated. So we'll see one after the other. One is the short interfering, another one is hairpin, another one is micro. This short interfering with double-stranded RNA about 19 to 23, and this one is about 4 to, with loop of 4 to 11, but 19 small loop you can see, and this one 20 to 24. Like this, differences will be there. Whatever may be, this is shRNA. If you see this one, this is short hairpin. It is introduced through the plasmid. Plasmid is a vector and entering directly into the nucleus.
Excuse me.
This is cytoplasm.
Excuse me, madam. Sorry for the interruption. Excuse me. Please need another 10 or 20 minutes, madam.
Break,
yes. So this is interrupted Zoom. Please, sorry for the interruption. All of you, please join through same link. Please join through same link.
Go through the slide. So these are the microRNAs. So how they appear? I will end up in 10 minutes, madam. Srivalli, madam. Transgene with hairpin structure and powerful inducers of RNAi. How they are interfering and how this one getting degraded. So these are the different types of mechanisms by different types of RNAis. So finally comes to what are the applications you can see. So there are so many applications, so much of research is going on for the last decade. So here RNAi has emerged as a novel alternative and provided to be potential for engineering of crops, for disease resistance and also pest resistance, for also agronomically important trends like nutritional quality. So any target gene either in the host plant or in the pest or in the pathogen effectively silenced. So they can be silenced by this mechanism. The genome sequencing projects, they facilitate for the identification of the target gene in pathogen and also in the pest. So this is one of the live example here. See, this is the insect. This insect is spoiling this particular plant. So what do you want to do? You want to get a plant, even though insect bites, this must be resistant. So how this RNAi is creating, that means this RNA, this is a genetically modified plant. Whenever it enters, the RNA of this particular insect is getting broken into small pieces. So it cannot survive, it cannot spoil the plant. This is one example. Next, pest control. Similarly, the pest corn rotworm cell, which is also published in one of the article. I have taken from that article. So next comes to the insect resistance. It is also transgenic plant. In that transgenic plants, If insect comes and again attacks that plant double-stranded RNA breaks the or silences the gene of the insect, which is responsible for spoiling. So that's the article also, that abstract is given. If anybody interested, can go through that one. Similarly, Gossipal. So similarly, so the plants that how they have roots damaging insects, all those things. Nematode resistance in transgenic plants. They also RNA is, our gene is silenced due to double-stranded RNA. So enhanced fungal resistance in the transgenic plants. They express a double-stranded RNA of gene that our gene pair ODC gene and related one. So again, the rice plant that is grown against, this is also published, rice fungal bacilliform virus, the same mechanism that RNA interference. So whatever genetically engineered plants are raised, all those things have work done for RNA interference also. Abiotic stress tolerant plants. So here also delta codeine, synthase gene, seeds expressing. So cotton contains much more seed than fiber by weight, rich in oil and protein. Some of that seed is fed directly to livestock and poultry, but the oil from the seeds must be purified before people can eat. So for that they made Gossipal free, so which is toxic to that one by RNA interference mechanism. For same way, indehiscence RNAi of the indehiscence gene result in seed shattering resistance. So these are the applications. So rosy outlook for blue roses. So creation of the blue rose now became very important for the breeders. So the transgenic rose developed by two companies, they are commercialized this product, Florizen and also Suntory. These two industries, they are working on this one. So there are 3 genes responsible for that. So those 3 genes they have isolated and they have developed particular blue rose using RNAi technology. So another application, improving lysine content, fatty acid content, decaffeinated coffee, narcotic coffee, tearless onions. Some people get tears while cutting the onions. So people started thinking about tearless onions to produce RNAi technology. So these are the different tomatoes, Solanum lycopessica. So all those things. Tomato that is resistant to Helicoverpa. So nowadays, including this RNA interference, they started creating artificial RNA-MI, that is a genetic construct they are preparing, that is called gene silencing by artificial miRNAs technology. This technique also nowadays famous. I'm not going in details now. So along with this technology, so there are this is a commercialization. So many plants tried gene. So how this is suppressed, the path suppressed, and regulatory approach, what is the phenotype that is resulted or expected. These are all commercialized base. So RNAi, that is RNA interference, is a bloom of biotechnology. So here, genome editing, that is a CRISPR, nowadays they are practicing, that is the CRISPR-Cas9 technology. Okay, thank you, sir. Thank you. I just went through.
Okay, thank you very much, madam.
Is it audible, my voice thoroughly, or any breaks?
Yes, madam. Your voice is clearly audible, madam.
Okay, okay. No disturbance.
Madam, Srivalli, madam.
Srivalli, madam.
Please unmute yourself.
I'm not audible, madam. Unmute. Madam, unmute. Madam, voice not audible.
Madam, please unmute yourself. Srivalli, madam.
Oh, okay, sir.
Madam, Please stop your share screen, madam. Madam.
Thank you, madam. Dr. Madhuri, madam garu.
Madhuri, madam garu. Please stop your screen sharing.
Thank you, madam. Thank you. A wonderful session by madam ji. Really very informative. Thank you for sharing your knowledge. You have covered so many topics, including crop improvement, genetically modified plants, markers, and especially RNA interference, very advanced technology. And we are lucky enough to have you today, madam. And students learn many new words like dsRNA, lysozyme, siRNA, guide RNA, and RNA-based therapy, RNA non-coding RNA, post-translation, and DNA silencing, and different techniques and difference between siRNA, shRNA, and miRNA, and blue roses, then artificial RNA. So many things we have learned today. Thank you very much, madam. Telugulo cheppalante chaala baga chepparu. Very happy today. Madam, I will call you personally. Thank you, madam. Now I request our participants, please any doubts regarding this session. Hello, sir. THIRUPATHI, sir.
Okay, madam. Okay, any queries, sir? Any queries? My lab mate and my eminent scientist, Dr. Gulab Khan, sir, is joined.
Sir.
Dr. Gulab Khan, sir. Dr. Gulab Khan, sir, from Kashmir. Central Silk Board from Kashmir. Dr. Gulab Khan, sir, has joined. Gulab Khan, sir.
Sir, we welcome you, sir.
Senior scientist from in Central Silk Board of India from Kashmir. Gulab Khan, sir, please welcome. Any queries? Any queries? Other participant, please unmute yourself. If you have any queries, please raise your hand or chat the box and message in the chat box also. If you are unable to speak, please you message post in the chat box. We will ask our respected keynote speaker. Okay, definitely we will send feedback link also. Shivalli Madam Garu, please send feedback link in the chat box.
Okay, sir. Let us conclude our session.
Okay.
Sir is there. Kashmir, sir.
Okay. So with the permission of our keynote speaker and dignitaries, we will go into wind up the session. Before wind up the session, we would like to express a few more words really regarding of today's national webinar. First of all, Department of Botany, both the institutions, we are wholeheartedly thankful to all the both the colleges patrons, Dr. Y. Satyanarayana Garu, Principal, Government Degree College for Women, JAGTIAL, and Dr. Pitla Das Garu, Principal, Government Degree College, Gambhiravpet, and conveners of the today's national webinar, Dr. P. Chandraya Sir Garu and Srivalli Bojja Madam Garu, and the organizing committee members, Dr. P. Bhikshamaya Garu, IKAC coordinator, GDC Gambhiravpet, and Dr. P. Krishnamurthy Garu, senior most faculty, GDC Gambhiravpet, and IKAC coordinator of GDCW JAGTIAL, Dr. Harjot Kaur Madam Garu, Vice Principal, Dr. Masroor Sultana Madam Garu, and NSS Program Officer Dharmapuri Garu, GDC Gambiropet, and all the respected faculty members of both the colleges, and all the delegates and all the dignitaries from different universities and different degree and PG colleges from various universities of our nation. We are wholeheartedly thankful to you for the grand success of today's national webinar. So, and also we hope this national webinar is definitely useful for our students and also our lecturer pattern, teaching fraternity also. And also we wholeheartedly thankful to our today's keynote speaker, eminent professor MADHURI.V Madam Garu. Wholeheartedly thankful to you because of your spare your valuable time, more than 2 hours from this regarding of this national webinar. And also thankful to all the teaching and non-teaching faculty of both the colleges and students also for grand success of this today's national webinar. And we expect from your side, your cooperation, your support and encouragement for conducting of this national webinars and workshops, and in coming webinars also we will going to plan for other new topics and new area of current scenario. So we are wholeheartedly thankful for your encouragement and supporting of all the faculties of both the institutions and participants. So we are wholeheartedly thankful to you for grand success of this national webinar. So I hope you are all filled this feedback link. No doubt at all, within 2 days or today by tomorrow, more than 400 participants, they are registering of this national webinar from different states of our country. So within 4 days or 5 days, you will get this online certificate through your registered mail. Definitely, if any queries or if you do not get the certificate, please you put WhatsApp. Definitely we will send your certificate. Thank you very much. And lastly but not least, all the botany faculty members and botany departments of both the colleges and keynote speaker, wholeheartedly thankful to you once again. With your permission, we will going to wind up this session. I hope you all get the feedback link. Participants, all the respected faculty members and students. I hope you, Madam, Malli okkasari meer link malli okkasari pampandi Madam.
Sir, please share.
Vallu malli share cheyamantunnaru. Malli okkasari meer feedback link pampandi.
Okay sir, I will share immediately.
Okay. So those who are filled the feedback link, so this session is over. All of you, thank you for your cooperation and encouragement. Krishnamurthy sir, thank you very much for your cooperation. Please unmute yourself. Krishnamurthy sir, please speak few words. Head Department of English, Dr. P. Krishnamurthy garu. Please.
Sir.
Sir, మాట్లాడండి. Please unmute yourself.
Sir, it's really a great thing that Padala Tirupathi Garu has taken a lot of initiative steps to educate the students, to upgrade the faculty members of UNIV and PG colleges. And before that one, I would like to say sincere thanks to the two patrons of the two colleges. So JDC Women, JAGTIAL, which is pioneering in the educational field like anything. And of course, no less than that college, JDC Gambiropet, is just galloping with so much of speed of achieving a lot of academic excellences in the nowadays under the chairmanship of P. Das Garu. So both the colleges under the chairmanship of Dr. Satyanarayana Garu and P. Das Garu, these colleges are creating wonderful success in all the areas that they have attempted. And for this one, really, Padala, Dr. Padala, THIRUPATHI Garu has been doing wonderful job. Not only this one, but he has been initiating wonderful steps and for creating areas and avenues to learn more about the subject, to learn more beyond the subject. And for this beautiful, wonderful, educative, informative program, here is the keynote speaker. Professor V. Madhuri Garu. What a wonderful information that she has given. I can't just keep the words, keep that one in the form of words. So all evidential information that she has presented, even the most difficult and radiant subject that has been kept in order in a systematic way, in a very easy manner. And our students really, they express a lot of thanks to you, madam, on behalf of the students of JDC, Women JAGTIAL, on behalf of the principal, staff, and students of JDC Gambhira. And really, it's a great privilege and honor for me to be a member in this participative webinar. And the students are expressing so much of acknowledgments to you, ma'am. Really, this is, though you are far away from this place, if you are to be here, maybe in Chatavana University or Usmania University, definitely will be coming very soon, maybe tomorrow itself. I think our college students and GDC women, JAGTIAL students may come and just talk to you. And you have been an inspiration, not only to the students, but also to the faculty members. And of course, convener, our Sridevi Madam Garu, convener, Chandra Garu, and convener and other co-conveners and other faculty members who have supported, and other technical staff members who have supported this one. I sincerely request our Dr. Satyanarayana Garu, the principal, and our P. Das Garu, the principal of JDC Gambhira, and other faculty members like organizing secretary like Padala THIRUPATHI Garu to initiate a lot of programs like that one. In that way, the students community will be benefited in a larger manner, though they cannot come and just study under your guidance, but they just definitely enjoy your teachings through online mode. Thank you very much. Thank you very much from the side of JDC Gambhira.
Okay.
Thank you,
Really,
thank you
it's
very
a
much.
wonderful, sir, your words are very inspiring and motivating us, sir. So definitely both the colleges, definitely we will going to associate to make more webinars and make more activities regarding of our higher education system. At the same time, also thankful to both the college principals. And Dr. P. KRISHNA Garu, especially wholeheartedly thankful to you regarding of your words, really inspiring us, sir. Thank you very much.
Thank you, sir. Thank you all.
So, madam, please once again, please share a feedback link. Some more participants is asking me.
Okay, sir.
So all the participants, please fill the feedback link. Then you will get e-online certificate through your registered mail, sir. So if you have any queries or if you have any problem for getting of e-online certificate, definitely you will post the WhatsApp. Definitely we will send within 2 or 3 days. Thank you for your cooperation. Thank you for your cooperation. And finally, Srivalli Madam Garu, wholeheartedly thankful to you because you are organizing the total entire session is smoothly and very effective manner. Thank you very much. And GDCW JAGTIAL, I hope on GDC Gambhira, definitely you will get good grade on and becoming forthcoming NAC process. Our senior host faculties like Dr. P. Garu and Dr. B. Pikshamaya Garu, education coordinator and head department of English. Krishnamurthy sir is also there. Definitely GDC Gambhira, rocking, good score in CGPA of NAC grade and also get B++. Definitely we hope.
Thank you, sir. Thank you.
Thank you, sir. Thank you for your support.
Okay, with your permission, we will going to wind up. Krishnamurthy sir and all the participants. Right.